Let's see how this works. Okay, so I may not be 100% correct about this, but I think I'm one of the few people that's thinking about this in non-mammalian models. So from my perspective, flies and humans are basically the same thing. Flies are just humans with wings. And that's because of the shared evolutionary ancestry. And that shared evolutionary ancestry makes flies an excellent model for studying some of these questions, such as obesity and metabolic syndrome. So before I get too far into this, I wanted to thank a number of people, including a large number of undergraduates. The first two columns there are largely undergraduate students who have contributed to the research that I'm going to be showing you today, as well as a number of collaborators and funding sources. So flies take a lot of work. So metabolic syndrome, I'm not going to go through the details here. It's complex. Bottom line, like as we've been talking about, calories in is greater than calories expended. That's the bottom line, but there's lots of complex physiologies underlying that. And within that, we have a set of symptoms which include abdominal obesity, elevated fasting glucose, dyslipidemia, and elevated blood pressure that are characteristic of metabolic syndrome. It's affecting greater than 30% of Americans and results in a 6-fold increase in cardiovascular mortality and a 5-fold increase in developing type 2 diabetes. But I'm an evolutionary biologist. So from my perspective, this is a maladaptive trait with genetic and environmental influences. So as an evolutionary biologist, I'm thinking, how can we have a maladaptive trait occurring in a population? So just a brief introduction to my study organism. Fruit flies go through, well, like everything, a developmental life cycle. I focus most of my research in those early stages, so the larval stages after the egg hatches. The first instar, I put the first instar larva onto various dietary treatments, let them develop through into either the third instar or pupal stage for most of the studies that I do. So flies and humans, as I mentioned, are basically the same thing. One thing is that flies get heart disease. So in the upper left there is a figure of a 1-week-old adult fly versus a 5-week-old adult fly. And as it ages, it develops arrhythmias, the same way humans have a tendency to do. Our central insulin signaling systems are virtually homologous, highly conserved. So all those mechanisms are largely the same. And then if you look at the fat body end of the fly, which is a dominant organ, that's essentially the adipose tissue, liver, and innate immune system of a mammalian system. So there's a lot of similarity there. Okay, so where does this phenotypic variation come from? In this case, variation in metabolic syndrome. We start out with our genome. That's transcribed into our transcriptome. That's translated into our proteome, which affects our metabolome, our set of metabolites, and that ultimately results in our phenome, the physiologies we're studying. But it's not as simple as that. We have influences from the environment affecting all of those physiological levels, and we have natural genetic variation that's contributing variation from the top down. And so I'm really interested in looking at the interaction between these two factors. So if we have a wild population of humans or fruit flies, and here they're showing their physiological diversity as a variety of colors. What I can do with flies, but not humans, is I can bring them into the lab and create inbred lines where I have many genetically identical individuals of each genotype, so I can represent the genetic diversity in that population, and from that, estimate the amount of genetic variation that contributes to the underlying phenotypic variation that was observed in the wild population. But there's also environmental heterogeneity, as we well know. This is where flies are an advantage over humans, because I can't do this to humans, but I can do this to flies, where I can expose exactly the same genotype to a variety of different environments and study then the interaction between the environment and the genotype and partition how important that interaction is relative to the individual effects of environment and genotype alone. So the basic experiment, I put 50 larvae from 20 genotypes onto these two diets, a normal diet and a high-fat diet, and then I measured their weight, triglyceride storage, and blood sugar. So possible outcomes, say for weight, if we compare each of these lines here is indicating a different genetic line. So it's possible that the individual genotype neither gains or loses weight on these two different dietary treatments. You probably know people like that can seem to eat whatever they want and are unaffected by their diet. But there might be some that are just naturally heavier than others for genetic reasons. But then there's some that are going to be sensitive to their diet that will specifically gain or lose weight in a diet-specific way. So those are possible scenarios we can see. Out of the 20 lines that were in this study, these are the various options we saw. And basically, when you start adding more lines, which I've done in other studies, it gets more and more complex. Basically, every possible phenotypic outcome, every possible reaction to diet is observed in a natural population. And so when we actually partition that variation that I was talking about, here this bar graph is showing in the red is the genetic contribution to the total phenotypic variation for the traits of weight, triglycerides, and blood sugar. The blue is the diet effect alone, and then the green is the interaction effect. So the takeaway message here is that diet alone is a minor contributor to the total phenotypic variation. That what's more important, much more important, is how diet interacts with genotype. So this might help explain all of these fad diets, some of which might work for some people. Many don't work for anybody. But almost always there's some kind of disclaimer at the bottom of those ads that say results not typical or results may vary. And that's in part because of the underlying genetic variation in human populations. The grapefruit diet might work really well for some people. That might be a great way for a few people to lose weight. But that doesn't mean it's just because it's good for one genotype, it's good for everybody else. So going back to this mechanisms of phenotypic variation, what I want to do, the next piece of what I'm looking at, is identify, if we have our phenome, how do the intervening physiological levels contribute to that? How does the transcriptome contribute to that? And how does the metabolome contribute to that? So going back to this basic experiment, we measure those original phenotypes of weight, triglyceride storage, and blood sugar. And then we also do whole genome expression profiling across all of these genotypes across all these different dietary effects. And we also do metabolomics. So we do GCMS profiling. In this initial study, we did 180 metabolites that could be well characterized. Those numbers are increasing now as metabolomics is a growing field, and it's improving. Basically, we found that the principal component of metabolite PC2, which is a breakdown or summary of the metabolomic variation that was present, was good at describing a number of physiologies, including body weight, so it was negatively correlated with body weight. If we look at the metabolites that are heavily weighted in metabolite PC2, they consist of L-DOPA and N-arachidonyl dopamine, which are, so L-DOPA you might be familiar with as a precursor to dopamine. N-arachidonyl dopamine was actually pretty, well, initially scary and then exciting to find out because it first showed up as THC in my analyses, which was really nerve-wracking. And then I realized it was probably an endocannabinoid. Endocannabinoids are known to play an important role in appetite. So this is actually the first known example of endocannabinoids playing a role in invertebrates in their physiology. Also included in this group are the branched-chain amino acids, which, if you have been paying attention, are often associated with metabolic syndrome and obesity traits in mammals. So this is, once again, evidence that these flies can be a good model of what's happening in mammalian systems. Finally, we measured the Arrhythmia Index, so I mentioned that flies get heart disease. And we found that this metabolic principal component was predictive of developing heart arrhythmias as the flies age. So these are all relevant physiological markers for the development of metabolic syndrome in the flies. So if we track down a few genes, just a handful of, just to give you an example, one gene we were interested in that was strongly correlated with all these traits was easily shocked. And we found that easily shocked is associated with both weight and glucose storage in our flies. So what we found in the population level survey was reflected in functional follow-up with that gene in our system. And then another gene, which has the catchy name of CG14332, which is how many genes are named in flies because we don't know what they are. So we have no previous knowledge of what their function is, but when we find that it was strongly correlated with these physiologies, and we can confirm with functional follow-up that it is indeed playing a role. So this is an example of how we can find new genes that are playing a role in metabolic syndrome by using flies as a model. So the current major focus in my lab is trying to actually genetically map QTLs that are, or genetic loci that are contributing to metabolic syndrome. We're doing this with a population that's based on 8 founders, so we have more genetic variation in the population than standard QTL mapping studies. And basically, we produce this population of recombinant inbred lines, where each line is genetically isogenic, but there's genetic variation from the original founders across the lines. Once again, we raise them on those 2 diets, a high-fat and normal diet, and then we measure the phenotypes, and from that, we can do association mapping to identify the positions in the genome that are playing a role in these traits. The statistical analysis I'm doing in collaboration with researchers at NCSU and their statistical genetics unit. So this is a basic map. What's going across the X chromosome, the X axis are the chromosomes. So the X, the second and the third chromosomes, the left and right arms are the second and third chromosomes in the fly. The Y-axis is the significance level for a given QTL, or given position in the genome. And here, just with the orange arrows, are indicating ones that reach genome-wide significance. If we compare the main genetic effects, which is just sort of you look at the population as a whole without thinking about diet, and then compare that to the effects that are diet-specific, one of the things that's intriguing is that they're not necessarily the same loci. So the loci that influence how an individual interacts with the diet is different from the loci that determine its average weight. So this particular one is for pupil weight. We find that pattern is consistent across all the phenotypes we measure, so that the genotype-by-diet interacting loci are fundamentally different from the main effect genetic loci. We also find that there are loci that are in common across traits. So here we have male and female weight and triglycerides. So there's one QTL there that's consistent across those. So that's evidence of genetic linkage between those traits. Interestingly, and this is consistent with the general patterns I've seen, blood sugar is often not as well correlated with triglyceride storage and weight as triglyceride and weight are with each other. So no linking genetic effects there. But when we look at the interaction effects, there are some linking QTLs between triglycerides and sugar, and then between the weight phenotypes. Finally, we've looked at these metabolites, and this is, I know, a very difficult graph to see anything on. Basically, the y-axis are all the metabolites that we measured in a more recent study. The x-axis, once again, is the chromosomes across the genome. And what I want you to notice are there's some positions in the genome that have a number of metabolites associated with them. Those metabolites, that's a genetic locus that's affecting metabolism as a whole, or across the whole metabolome. Then we have other loci, or other metabolites that are associated with many positions in the genome. So those might be upstream effectors of metabolism as a whole. So we can actually map genetic loci that are influencing these traits. So some of the next steps, so far I've done diet and exercise, genotype and diet. And I spent a lot of time trying to think about how could I exercise flies. And we figured it out. We've invented this thing called the tread wheel. This talented undergraduate, Sean Mendez, actually built the prototype. And basically we haven't figured out how to exercise larvae yet, but we are exercising adults. And basically flies have a desire to go up when they're at the bottom of something, negative genotaxis. And so basically what we do is we put them on this hot dog turner in their vials that you can see there on the upper right. And those vials turn slowly, and so up keeps changing, and the flies keep walking. And we can run them for 30 minutes at a time for 3 rounds of 30 minutes at a time for a week. And then we're assess them at the end of that week, and basically have exercised fit flies. So, the next step of what we're going to be doing is whether we can use adult exercise to counteract juvenile diet. So if we give the flies a crappy diet as larvae and then exercise them as adults, can we counteract those negative effects? And then ultimately going to be genetically mapping those interacting effects as well. So the bottom line, genotype-by-diet interactions are a substantial contributor to metabolic syndrome variation in flies and therefore humans. Loci for genetic effects are different from those for genotype-by-diet interaction effects. Drosophila are an excellent model for the mechanisms of genotype-by-diet interactions underlying metabolic disease in humans. And ultimately, prevention methods will be most effective when we can match them to individuals' genotypes. So I think that's everything I had.
Hi, Sherry Barkin, Vanderbilt. First, I'm so delighted to know that I'm really closely related to a fly because it keeps my hope of flying alive. So thank you for that. And you know what strikes me as I hear about your incredible, thoughtful, systematic work is we know that the genotype hasn't changed much over time. So clearly when we look at that diet genotype interaction, I hope that you're going to be able to answer the questions about what in the diet. And I know we have to go beyond calories and macronutrients. So given that you're looking at metabolites, and given that our food and the constitution of our food has changed pretty dramatically, what insights do you have about that?
So I've also looked at sugar content and actually the types of sugar. So we've tried the amount of sugar and the types of sugar varying sucrose, glucose, fructose, and basically the type of sugar matters for the flies as well, as of course the quantity. And basically sucrose is the worst for them, and fructose is better, all else being equal. But yes, certainly the transition in the types of sugars in our diet might be playing an important role. And one other layer that we've been playing around with is the form of the fat. So the fat we've been using in most of these studies has been saturated fat from coconut oil. But I have a student who's working on testing different fats, including olive oil and walnut oil, to see if the unsaturated or undersaturated fats make a big difference, and they do. So the flies that eat olive oil instead of coconut oil are healthier than the ones that are, So it makes a difference what the source of the fat is. The one thing I was going to say, but yeah, I think that more or less covers it.
Claude Bouchard, Pennington Biomedical Research Center. That was a nice introduction to my talk. But I have two questions, many more, but two. The genotype effect on body weight, is that a large effect size?
Yes, yeah.
Like?
So about 25% of the total variance is due to genotype alone.
So that's irritability, about 25%.
Right, yeah.
The QTLs that you have identified, are they really meaningful? Do they have a large effect size or just?
Individually, they don't, no. Like in any one of these genome association type studies, a given QTL is 1 to 5% of the total variance, but collectively they do add up to a large portion of that total heritability. So.
And do you recover under these QTLs any of the genes that are known to be associated or to cause obesity in humans?
Not yet. So we have many candidate genes, and we're working on following up on those now. So far, the ones that looked most promising, like we had one gene named Pudgy, that looked like a very exciting one, but it didn't actually fall out with the association, with the functional follow-up. But yes, we're systematically working through those now. Thank you.